And this quote states, "Where a cell exists, there must have been a pre-existing cell, just as the animal arises only from an animal and the plant only from a plant." So what we're looking at here is that if we need new cells produced, they are going to come from cells that we already have. So it's somewhat like the chicken and the egg situation, where to produce cells, we need a cell, but then that cell must have come from another cell, and so on. Now, there's lots of reasons that we do have the cell cycle, or what we would call cell division, take place. One reason is going to be for the process of reproduction. And when we talk about the process of reproduction, this is going to be involving the production of egg and sperm cells, because egg and sperm cells are what we use for reproduction. And that is what we're going to look at primarily in chapter 13, so the next chapter. Another reason for cell division is for growth and development. We initially start as one fertilized egg, and that fertilized egg is going to divide many times to give us all of the cells that make us up as an adult individual. Tissue renewal will be yet another type of, or another role of cell division. We are constantly replacing cells. So this is due to cells that have been damaged or cells that are old. As our hair grows, we're replacing those hair cells. As if you donate blood, we're going to replace those blood cells. We continually lose skin cells. And so growth and development, tissue renewal, those two roles are really what we're looking at in this chapter, in chapter 12. Those are things that are taking place all of the time inside our bodies. So we are constantly growing and developing in some way, and we're constantly replacing some tissues. So those are going to be taking place all the time and pretty much all over our bodies as well. Now, as we start talking about cell division, it's important to understand that if we're going to have cell division take place, in other words, take one cell and split that one cell then into getting two cells, we're going to first have to copy all of the information in that original cell. So we're going to have genome or DNA duplication take place. The genome is the cell's endowment of DNA or genetic information. That's one way that we refer to it. You could also just say all of the DNA inside of an organism. When they talk about genome projects, like the Human Genome Project, that is just a project that goes through trying to figure out what all the DNA inside a human cell says, or what its sequence actually is. The DNA that we have is going to be primarily located in the nucleus, so the vast majority of it will be in that location, and that's what we're going to focus on in this chapter and then also in the next chapter. The DNA in those locations is a double helix, but it's going to specifically be a double helix that is a linear molecule. That DNA molecule is going to be in the form of a chromosome. Chromosomes are going to be made up of DNA and some associated molecules, which are proteins, and together that DNA and the associated molecules are going to be called chromatin. That chromatin is going to help us to really organize the DNA molecules inside the cell. Now, if we talk chromosome numbers, every species has a characteristic number of chromosomes. Humans, if we look at humans, they are on this diagram. Humans have 46 chromosomes. So that means I should have 46 chromosomes. You should have 46 chromosomes. If we look at other animals, if we just stick with the animal side for a moment, fruit fly has 8 chromosomes, so a rather small chromosome number, which makes it an easy organism to use, or fairly easy organism to use, if we're trying to do genetic studies, because there just are not as many chromosomes there. Rabbits have 44. We can see that cows have 60, dogs have 78, and so on. One thing that we want to point out here is that there does not appear to be a real correlation between the number of chromosomes that an organism has and how complex that organism is. Now, it's hard to really rank the complexity of organisms, but as humans, we like to think that we are the most complex of all organisms. But as you can see from this table, there are many organisms that have higher chromosome numbers than humans. So it does not appear that there really is a strong correlation there between number of chromosomes and complexity. If we look at the plant side of all of this, I want to point out the first plant that we see on this list, which is Arabidopsis thaliana. That plant you may not have heard of before, but it is a very important plant when we start talking about plant research. That organism's genome has been sequenced, and it is used for a lot of experiments, really a starting place for experiments in the plant research community. As you can see, plant chromosome numbers are comparable to what we have in animal species. Oftentimes, they actually are higher than what we have in animal species. The fern that we see on the bottom of the list has the highest known chromosome number of any species. So in that fern, there are over 1,200 chromosomes in every single nucleus of every cell. So plant chromosome numbers can obviously get quite high. You don't need to memorize any of these chromosome numbers with the exception of the human chromosome number. But we do want to know that every species is going to have kind of a set chromosome number. Now, if you look at these numbers, if we talk about the chromosome structure, the average human nucleus size is about 0.0002 inches, so very tiny. The length of the DNA that's packed inside that cell is about 70 inches. So in every single nucleus, we're taking approximately 70 inches of DNA and we're packing it into 0.0002 inches. Seems like an impossibility that we would be able to take a molecule that is so long and pack it into such a tight, confined space. But this is something that is done inside every one of our nuclei and every single one of our cells. The way that this is going to be accomplished is very similar to a spool of thread that is used for sewing. Those spools of thread have lots and lots of yards of thread on them. If you ever take a look at one of those spools, and those spools are maybe 1 inch by 2 inches, they have 500 yards on average on top of one of those spools. So 500 yards, we're talking about 5 football fields. So that thread is able to be packed in such a tight little spool or small spool because it is such a thin molecule. And not only that, but we're going to wrap it in a very organized fashion around that spool. The proteins that are associated with DNA are going to do essentially the same thing for the DNA molecule. So what will happen if we look on here is that the DNA, which will be a double helix, is going to be wrapped around these special proteins in a very organized fashion. So we've got looping around these, very organized. So we have about 2.4 loops of wrapping, a specific distance between the spools that we have. We will have additional proteins that will act as kind of a scaffold that we can start to wrap or twist these around. We will have additional proteins that will enable looping to take place, and then we'll get more coiling, and eventually we will get to a chromosome shape that you have probably seen a picture of at some point in your life. And that is the typical X-looking chromosome. When we see a chromosome that looks like an X, what we're really seeing there is not just DNA, but it's DNA wrapped up with a whole lot of proteins. And in fact, it's highly wrapped up around a lot of proteins, such that it's really quite difficult to get to the DNA to read it when it's in that form. This means that the DNA on an everyday basis will not be packed up to that extreme. Instead, it'll be somewhere between highly packed up and completely unpacked. So somewhere in the middle between the two. There will be specific times during the lifetime of the cell when it will want to have the DNA pretty much all opened up. There will be other times when it will want to have it pretty much all highly compacted or coiled up. So we'll talk about those times. Here we see a typical nucleus. Inside this nucleus, it does look like a tangled mess, but keep in mind that that DNA, where we're looking at it there, it's not highly compacted. It's going to be somewhere between the two extremes as we were talking about. But even so, it is going to be organized, and a lot of that organization is due to the nuclear lamina that's going to be just inside that nuclear envelope. So although we look at that picture and it looks like a mess, there is organization there. We talk about the chromosome structure. When we look at a chromosome, you've probably seen pictures of chromosomes that look similar to what we're looking at right here. The chromosome does have this central area, which is what we tend to call a centromere. So the centromere is a little pinched in area. It may or may not be at the center of the chromosome. The ends or the tips of the chromosome are going to be what we call telomeres. So in this picture, we're actually seeing 4 telomeres. So we have a tip here, here, and then we've got 2 telomeres down here. And then we also have arms. The arms are the individual pieces that we see here. There are 4 arms. There are no legs when we're talking about chromosome structure. So when we look at that chromosome, we're seeing DNA wrapped up a whole bunch around lots and lots of individual proteins. Now we're going to be talking about 2 different types of cells. These are somatic cells and then reproductive cells. In the somatic cells, or with the case of somatic cells, another way to refer to these would be to call them vegetative cells. This is in contrast to what we call the reproductive cells or the gametes. The reproductive cells have many other names. Sometimes they're called sex cells. Sometimes they're called germ cells. But what we're really looking at with the reproductive cells is this is going to be either sperm or it will be egg. So when we define the somatic cells, it's easy just to say that all of those cells that are not the sperm and the egg. So somatic cells in the case of a human would include skin cells, hair cells, blood cells, nerve cells, all of those types of cells. In this chapter, we're going to talk about how we produce the somatic cells. When we produce the somatic cells, initially the DNA will have to be duplicated. So that is going to take place. As we see in this picture, we're going to start with a cell that has DNA that has not been duplicated or not been copied. That DNA is going to be composed of linear double helixes, and we're going to go through the process of copying all of that DNA and making an exact copy of that DNA. When that happens, we're going to end up with 2 molecules of linear DNA. So notice we've got 2 of them here, and if you compare that to what our chromosomes are looking at on this side, notice that at this point we're seeing that chromosome that looks like an X. Okay, the one that we're most familiar with seeing. That is not how the chromosome really typically looks inside the cell. Up here on the top, we're seeing a chromosome that just looks like this, and I'll put a little dot there to represent the centromere. Most of the time, the chromosome is going to have just that one double helix piece there. When we look at it in the form of an X, that's a chromosome that actually has twice as much information there as what we really need. And it has that because we've copied all of it just recently so that we could ultimately divide that out and put a copy into each of these 2 new cells that we're going to be producing. If we talk some terminology, anytime we talk about one piece of DNA or one linear double helix, that's what we call a chromatid. Here we're seeing 1 chromatid. Here we see 2 chromatids, because notice we've got two linear pieces of double-stranded DNA. Down here on the bottom, we're also seeing 2 chromatids total. Now, it is possible for chromosomes to be composed of 1 chromatid or to be composed of 2 chromatids. Up at the top, we have basically 1 chromatid and 1 chromosome. Okay, so those are the ones that do not look like the X shape. This middle picture has 2 chromatids, but it is only 1 chromosome. Notice that it looks like an X. This is what we a lot of times call a replicated chromosome. We could also call it a duplicated chromosome. Essentially, all of the content has been copied so that we will have the extra copy to put into this new cell that we're producing. Down here on the bottom, if we look at this, we do have 2 chromatids here, but really we have 2 chromosomes with 1 chromatid each. Okay, so the difference is a chromosome is 1 basically piece. That piece of DNA wrapped around proteins may be composed of 1 DNA molecule, it may be composed of 2 DNA molecules. And the DNA molecules are the chromatids. We do want to understand that terminology because we will be referring to it quite a lot throughout this chapter and then also the next chapter. Okay, so we can have chromosomes composed of 1 chromatid each, which is what we see here, here, and here. Or we can have chromosomes composed of 2 chromatids each. And that's the ones that we call replicated or duplicated. Now, if we look at this more, when we go through the process of DNA replication, which we will look at in a later chapter, chapter 16, we will be taking this original copy that we have and we will be basically building that second half right on top of it. When we do that, these 2 are going to be identical to each other. Identical because we're taking 1 and we're building the second half right on top of it, an exact copy. These are what we refer to as sister chromatids. So how the sister chromatids compare with each other is they are going to be identical in every possible way. That means that if we later separate them out into these 2 new cells, those 2 cells should have exactly the same genetic content in them also. The process of cell division in a eukaryotic cell is 2-part. We're going to have mitosis, which will be division of the nucleus and all of the content inside the nucleus. And then we also have cytokinesis, which is going to follow this up, and that will be division of the cytoplasm, or in other words, the rest of the cell, so not the nucleus. When we take these 2 and we combine them together, we have mitosis and cytokinesis. That will then give us our somatic cells. Somatic cells are going to be cells that are genetically identical in every way. Mitosis and cytokinesis produce somatic cells, they produce identical cells, and all of our somatic cells did come about through the process of mitosis and cytokinesis. When we have mitosis and cytokinesis take place, we're going to start with 1 cell, which we will call a parent cell, and we're going to take that cell and we're going to split it into 2 new cells, and those new cells will be what we call daughter cells. Now notice we're going to be splitting or cutting that parent cell in half, so we will no longer have that parent cell. If we have mitosis and cytokinesis take place again, we could split each one of these daughter cells into 2 more cells, and again, those would go away. So we start essentially as a fertilized egg. So this is us right here at the very beginning. This could be your embryo, as you call it, and that egg is going to divide many times by mitosis and cytokinesis to produce all the cells that you now see inside your body. That means that every single one of your cells should have exactly the same DNA in it. This is really why the forensics sciences actually work. Because it doesn't matter if they get a blood sample or a hair sample or a skin sample, saliva, you name it, every one of those cells should have the same DNA. So if they have that, they should be able to match that DNA to a particular person. Now, if we go on to talk about how this process is going to take place, we want to look at the overall life cycle of the cell. When we look at the life cycle of the cell, there really is just one tiny little portion here that is the mitosis and cytokinesis. So that would be what we would call the mitotic phase or the M phase of the cell cycle. Notice that what is not M phase falls into this big category which we call interphase. Interphase takes up much more time than the mitotic phase does, and it can be divided into 3 different phases. So we're going to look at each one of these phases and talk about what's happening in the phases. But we want to understand that the purpose of interphase is to prepare for the mitosis and cytokinesis. So that is the overall goal. We want to basically copy everything inside that parent cell and make sure that we're ready to divide. So if we look at the first phase of interphase, this is what we call G1, and the G stands for gap 1. In gap 1, we are dedicated to copying everything inside the cell except for the chromosomes. Okay, so we're not copying the DNA at this point, but we're copying any enzymes that are there, we're copying organelles, we're copying cytoplasm, membranes, all of those other things. So that happens during G1. If we're looking at a cell that is in G1, the chromosomes are going to be composed of 1 chromatid each. So if I just draw a cell here that has 2 chromosomes, we've got chromosomes composed of 1 chromatid each. Once we get done with gap 1, we're going to move into S phase. The S of S phase stands for synthesis, because this is when the DNA is actually being copied or replicated. So when we're talking about S phase, we're going to start with cells that have chromosomes that look like this. And then throughout the middle here, really what we're doing is we're building that second half so that by the time we finish, we now have those chromosomes that look like X's. So we have copied them and we have made the sister chromatids. Now we've got in this picture 2 chromosomes composed of 2 chromatids each to give us a total of 4 chromatids. G2 stands for gap 2. And gap 2 is really a double checking phase. So in gap 2, we are double checking everything that we've done in gap 1 and also what we've done in the S phase. We want to make sure that we have everything prepared and then we've got our DNA copied exactly so that when we do actually divide and produce our 2 daughter cells from this, we are going to get those cells that are genetically identical to the parent cell, and also we want them to be identical to each other in every possible way. If everything checks out okay in G2, then the cell will get the signal that yes, it can go ahead and divide by mitosis and cytokinesis. So notice that a whole lot of time is spent on the actual preparation for the cell division, but then the actual cell division part doesn't take all that long. Now if we talk numbers, this is a look at a typical human cell cycle. Notice that we're going to spend about 10 hours in G1, so copying everything in the cell that is not DNA. We're going to spend another 9 hours copying the DNA specifically, another 4 hours double checking everything, and then we're going to spend 1 hour on the actual splitting process. This is a 24-hour cycle. Notice that out of 24 hours, 23 of those hours were spent in the preparation part. 1 hour was spent in the actual dividing. So most of the effort is during the interphase. The mitosis is a very fast process.